Hi everyone and welcome in my channel Age Life Science. Here I am talking about the differentiating the pathways for antigen presentation by MSc class first and MSc class second molecule. So please subscribe my channel Age Life Science, like my videos and share it to your friends, and don't forget to press the bell icon so you will get the notification of my newly uploaded video. So let's start. Here in this image, the some differentiating characteristics by MHC class first molecule and MHC class second molecule for antigen presentation are listed. The first feature, a composition of a stable peptide MSC complex. The This MSC complex or a class first MSC molecule is expressed by all nucleated cells, and this is made up of alpha and beta chain. The alpha chain is made up of alpha 1, alpha 2 and alpha 3 domain, and beta chain is made up of a beta 2 microglobulin domain. This is a loaded antigen or a loaded peptide antigen, and this will be flagged or this will be shown to the CD8 plus cell or a T cytotoxic cell, and then after expression of this peptide antigen by a class first molecule to the CD8 plus cell, they will interact with a alpha 3 domain of a class first molecule and makes a binding, and after binding they carry out their reactions by a different ways. They secrete granzymes, they secrete cytokines, and otherwise they makes a call to the remaining cells of the immune system and carry out a total degradation of that infected cell or a tumorous cell. This is action carried out by the that CD8 plus cells. For MHC class second molecule, the MHC class second molecule is made up of two chains, alpha chain and beta chain. The alpha chain is further made up of two domains, alpha 1 and alpha 2. This is alpha 1 and this is alpha 2. Then beta chain is made up of two domains, beta 1 and beta 2. The alpha 2 domain of a class second molecule get interacted with a co-receptor of a CD4 plus cell or a T helper cell and binds with them. After binding of a T helper cell to this class second molecule, they will carry out their reaction. They identify which kind of antigen it is and which reaction have to take place, and they make a call to other remaining cells and carry out their actions. Then the second feature, types of APC. The class first molecule is expressed by all nucleated cells, whereas type second is expressed by a antigen presenting cell like dendritic cell, B lymphocyte, mononuclear phagocytic cell, endothelial cell, and thymic epithelium. Then the next point, responsive T cell. The antigen presented by a class first molecule will be responded by a CD8 plus cell or a T cytotoxic cell, whereas the antigen presented by a class second molecule will be responded by a CD4 plus cell or a T helper cell. Then next point, that is the source of protein antigens. For MHC class first molecule, the antigen is a endogenous. A virus antigen or a viral antigen or a tumor cell itself acts as a endogenous antigen. So they are processed within a cytosol and they are processed by a proteasome. In case of MHC class second molecule, the foreign antigen or an antigen which is outside of a cell that is a exogenous antigen engulfed by a process of phagocytosis through a endosomal and lysosomal vesicles. The endosomal and lysosomal vesicles, they carry out their reaction and process that foreign antigen after engulfing it. The next point, enzyme responsible for a peptide loading of MSC. The cytosolic proteasome are responsible for a peptide antigen loading for a MHC class first molecule, whereas the endosomal and lysosomal proteases, for example cathepsin, are responsible for a peptide loading on a MHC class second molecule. A site of peptide loading of MSC. The peptide loaded in an endoplasmic reticulum in the case of MHC class molecule, MHC class first molecule. In case of MHC class second molecule, it is loaded in a specialized vesicular compartment. Then the molecules involved in a transport of peptide and a loading of MSC molecule. The chaperone, transporter associated antigen presenting protein, that is a TAP protein, which are already present in a ER, these are involved in a transport of a peptide antigen and loading it into the MHC class first molecule. Whereas the chaperones in ER, invariant chain in ER, Golgi, these are responsible for a transport of peptide and loading of an antigen on a MHC class second molecule. Here in a one image we can look for two pathways, pathways presented by a class first molecule and class second molecule. This is only for your understanding. Let's starting from a class first molecule. Here it is a class first molecule. One second. A class second molecule with a loaded antigen, and here a class second molecule with a loaded antigen. This is expressed by a nucleated cell, this is expressed by a antigen presenting cell. This is flagged or shown to the CD8 plus cell, this is shown to a T helper cell or a CD4 plus cell. And this is the final step, and it's started from these steps. The first step, the virus or viral particles enters. No, this is a first step for a MH class first molecule. The viral antigen enters in a cell cytoplasm. With a ubiquitination, they are cleaved to a proteasome into a smaller fragments of the peptide or a smaller antigens, and with the help of a TAP protein, they are loaded on a MHC class first molecule, and this MHC class first molecule is a glycoprotein, and it is synthesized within a lumen of endoplasmic reticulum. After synthesis of it, the TAF loads the peptide antigen on that MHC, and this complex MHC class first molecule with a loaded peptide antigen is then transported to a Golgi complex through a vesicle, and vesicle through the cell surface receptor, and after it shown to this way. And this is a endogenous pathway which is represented here in a green. Towards the right, the foreign antigen or antigen which is present outside the cell or outside the APC, it is engulfed by a endosomal or a lysosomal vesicle like this. And within lysosomal vesicle, it is fragmented into a short peptide antigen. Simultaneously, the lumen of endoplasmic reticulum synthesizes a MHC class second molecule, and during this, the peptide binding group, it is occupied with a invariant chain protein, and this invariant chain with a class second molecule is then transported to a Golgi complex through a vesicle. Then Golgi to another vesicle. During this, the invariant chain gets degraded or digested, but remaining a small portion of a protein called as a clip class second associated protein, and this clip is useful to avoid the loading of endogenous antigen present in this cell. And when this endosomal vesicle with foreign antigen or a foreign peptide antigen and this MHC class second molecule with a clip, they are overlapped in one vesicle, and during this overlapping, the clip is removed with a peptide antigen. The MHC class second molecule's group is occupied with a peptide antigen, and from this through a vesicle, they are presented in this manner to the CD4 cell. This is the way by which the class second molecule express or shows the antigen to the CD4 cells. Here once again one clarification: a cytosolic pathway or a pathway by a MHC class first molecule or a endogenous pathway. In this pathway, the endogenous antigen or a viral antigen or a tumor cell antigen is ubiquitinized and ready for a cleavage through a proteasome complex, and this ubiquitinization is a ATP requiring process. After cleavage through a proteasome, the peptide antigens are formed, which are smaller in size. With the help of TAF, the TAF help for loading of this peptide antigen on a class first molecule. In the endogenous endocytic pathway, the exogenous antigen are processed through a or engulfed through a endocytosis or a phagocytosis by a endosome, and during this engulfment and in that vesicle through a number of hydrolytic enzymes, that exogenous antigen is converted into a smaller fragments of peptides, and these peptides are loaded on a MHC class second molecule. So thank you friends, thank you for watching me. So please subscribe my channel Easy Life Science, like my video and share it to your friends, and don't forget to press the bell icon so you will get a notification of my newly uploaded video.